Dear, dear students, Assalamu alaikum. Today we are going to study about the process which is called as cell cycle. As all of you know that living organisms are made up of cell. Cell is the basic structural and functional unit of the living organisms. Living organisms needs more and more number of cells in order to grow, in order to repair their damaged body portion, in order to form the new structures. All these new cells are produced as a result of cell cycle. So cell cycle can be defined as the series of events through which one cell divides to produce two or more than two new cells. Cell division is a complex process. So this process is further divided into two main steps. First one is the interphase and second one is the mitotic phase or M phase. Today we will study about the interphase. Inter mean in between two consecutive cell divisions. So interphase can be defined as the phase between two consecutive cell divisions. For example, look at this. This is a cell. This cell divides to produce two daughter cells. These daughter cells are further going to divide. Before division, this cell undergo a process which is called as interphase. Previously, it was considered that interphase is a resting phase, but after research, it was noted that it is not a resting phase, it is metabolically active phase, in which cell metabolically grow, accumulate different types of material, synthesize different types of substances in order to facilitate the next step, which is called as M phase. Interphase is further divided into three main steps. First one is the G1 phase, second one is the S phase, and third one is the G2 phase. These changes are involved during the process of interphase, which prepare the cell for the process of cell division. For example, look at this. These are the two cells which are produced by previously one parental cell, and these two daughter cells are going to divide further. For this purpose, first of all, they will pass through the changes which are called as G1 phase. It is the first step of the interphase. In G1 phase, there is the formation of cell organelles, synthesis of proteins, accumulation of materials, increase in the cell size, which is also called as growth. These are the main changes which involved in the G1 phase, and these changes are helpful to the cell for further division. Actually, in this phase, cell accumulate different types of material, for example, proteins, carbohydrates, lipids, etc., in order to form the new components, new organelles. Because these organelles which are present in these cells are half in number, because these are produced from this parental cell. This parental cell, when divides to these two cells, they have half the number of organelles, they are smaller in size. So these changes help the cell to produce different components to increase in the size. For example, look at this. This is the cell which is of greater size as compared to these parental cells. So these all changes takes place during the G1 phase. After G1 phase, some cells undergo a phase which is called as G0 phase, in which cell temporarily or permanently stop the division. For example, neuron cell, they undergo complete or permanent stoppage of cell division. Some cells, for example liver, kidney cells, etc., they will undergo the process of division after long time. Some cells do not undergo the G1 phase. They directly go to the next phase, which is called as S phase. During S phase stands for senses. Senses of what? Senses of DNA. During this process, replication of DNA takes place. Replication or duplication of chromatids takes place. And third one is the growth. So these are the three main changes which takes place during the S phase. The main event, the main change is replication of DNA. All of you know that DNA is the hereditary material. Before cell division, this hereditary material must be duplicated in order to equally distribute the genetic material in the daughter cells. For example, this is the cell having chromosome. This is the cell after S phase. This cell have duplicated its chromatids. These are called as sister chromatids. These chromatids increase in number in order to equally distribute the genetic material in the M phase or dividing phase. The next one is the G2 phase. G2 phase involves more growth. For this purpose, they accumulate more and more material. Next one is the synthesis of protein for the formation of spindle fibers, ester fibers, centriole, etc. Because these things are involved in the next phase, which is called as M phase. So these main changes takes place during the G2 phase, which actually prepare this cell for the process of cell division. Now this cell is ready for the next phase, or the phase which is called as M phase or mitotic division phase. This mitotic division phase or M phase is further of two types. One is mitosis and second one is meiosis. About these main steps we will study in next lecture, inshallah. So this is all about for today. Today we have studied about the main process which is called as cell cycle. Cell cycle involves two main phases which are called as interphase and mitotic or M phase. Interphase is further divided into three main steps. First one is the G1 phase, second one is the S phase, and third one is the G2 phase. And after these phases, this cell undergo or will undergo in a new phase which is called as M phase. So these are the main changes. So these are the main events which are involved in the interphase process. So this is all about for today. Inshallah, in next lecture we will study about the mitosis. Allah Hafiz.